Have you ever wondered what goes on inside your body? Have you ever wondered how you grow? One of the answers to both of these questions is mitosis, a type of cell division. Mitosis is essential for the replacement, development, and growth in eukaryotes. But how does it work? Before a cell is ready to undergo mitosis, it must go through a process called interphase. The cell grows, develops, and replicates its DNA to prepare for mitosis in this phase. Once a cell has completed interphase, it is equipped to begin mitosis. The first and longest step of mitosis is a process known as prophase, wherein the genetic material, DNA, condenses into chromosomes. During prometaphase, the next portion of prophase, the nuclear membrane dissolves and structures called centrioles move to the opposite cell poles to organize the spindle, an intricate network of fibers forming a protein structure that divides a cell's genetic material. The second phase of mitosis is metaphase. In this phase, all the chromosomes align at the center of the cell. The metaphase plate and the spindle fibers attach to the centromeres, which are the protein centers of the chromosomes. Next, the chromosomes are pulled apart into sister chromatids in a process known as anaphase. The sister chromatids are pairs of identical copies of DNA joined at their center by the centromere. The newly split sister chromatids are then pulled to the opposite sides of the cell, effectively separating it. The final phase is telophase, wherein the interior of the cell isolates into two portions with the same number of chromosomes, organelles, and membranes, separated at the cleavage furrow. This phase also includes the chromosomes unfolding back into the chromatin and the reformation of the nuclear membrane. This one division produces two somatic or non-sex cells that can be used for multiple purposes in eukaryotic organisms. The cells produced here are diploids, meaning they have an identical chromosome number with both sets of chromosomes and an identical genetic makeup to their parent cell. This guarantees the cells produced will be identically productive to their parent cell, as well as that efficiency in cells cannot increase with successive generations. As a result, there is a lack of diversity across the cells. However, this process does not apply to sex cells, so how do they divide? Gametes, or sex cells, divide through a process called meiosis. Meiosis is divided into two stages, meiosis 1 and meiosis 2, with each stage containing the same four core steps of mitosis. The chief difference between the two parts of meiosis is the number of chromosomes present at each stage. The first stage of meiosis is prophase 1. Just like prophase and mitosis, prophase 1 contains the same centrosome movement, spindle formation, and nuclear membrane breakdown. However, the similarities end there. Meiosis features a unique sub-process in its prophase 1 that is called crossing over. Crossing over is the process where the chromosomes given from each parent are broken down by proteins and interwoven with each other, resulting in genetic variation. Crossover occurs in an X-shaped region called the chiasmata. The next step is metaphase 1. This stage is very similar to metaphase and mitosis. The homologous chromosomes are aligned at the center along the metaphase plate, with each chromosome of the homologous chromosome pairs facing the opposite pole of the cell. However, unlike mitosis, the spindle fibers only attach to each chromosome at one point, and each pair is lined up independently of other pairs, which is a concept known as independent assortment. The third phase is anaphase 1. In this phase, the proteins responsible for the cohesion of the sister chromatids are broken, allowing the spindle to pull the homologous chromosomes towards opposite sides of the cell. The last stage of meiosis 1 is telophase 1 and cytokinesis. The cell divides into two haploid cells, but because of crossing over, some sister chromatids have regions of non-sister chromatid DNA. In animal cells, a cleavage furrow starts to form, and in plant cells, a cell plate forms, just like telophase in mitosis. In certain organisms, the chromosomes unfold and the nuclear membrane forms. The first stage of the second part of meiosis, meiosis 2, is prophase 2. Here, a spindle forms, and later on, the spindle starts moving the chromosomes to the metaphase plate. The next process is metaphase 2, where the chromosomes are positioned at the metaphase plate again with the help of the spindle. And because of crossing over, the sister chromatids are not genetically identical. The third stage of meiosis 2 is anaphase 2. In this phase, the proteins holding the sister chromatids in the centromere break down, allowing the spindle to separate the chromatids. The last stage of meiosis is telophase 2 and cytokinesis. The nuclei reform, the chromosomes decondense, and the cytoplasm divides. These two divisions produce four haploid daughter cells that are genetically different from one another and from the parent cell. The cells produced have half the number of chromosomes as the parent cell. The meiotic daughter cells are sex cells utilized for sexual reproduction, or reproduction that requires two parents. Meiosis, as opposed to mitosis, allows for genetic variation, so every cell is unique. These differences in DNA combinations across generations result in organisms having the potential to exhibit improved qualities when compared with their parents, but they also have the chance to inherit harmful characteristics compared to their parents. Therefore, the next time you wonder what is happening inside your body, be aware that mitosis and meiosis are taking place.